Our next presentation is Capstan Therapeutics, and I'd like to welcome Adrian Bott, Chief Scientific Officer and Executive Vice President of R&D, to the stage.
Thank you very much. We spend a lot of time discussing about cell therapy and cell manufacturing, but let's switch gears to talk about cell therapy without cells. So Capstan Therapeutics is a startup based in San Diego, focused on the concept of in vivo reprogramming of the immune system. And we have brought together several technologies. Most importantly, chimeric antigen receptor concept, together with our scalability using targeted lipid nanoparticles loaded with mRNA as a payload format. And to that aim, we have started our operations in close partnership with UPenn scientific founders, Carl June, as well as Drew Weissman and John Epstein, bringing together the targeted lipid nanoparticle technology, RNA chemistry, and our own focus on ionizable lipids, as well as chimeric antigen receptor design. Let's talk a little bit about our platform technology. We started with conventional lipid nanoparticles, the type of particles that are widely used for COVID-19 vaccines, that are redesigned or optimized at 3 different levels. Number one, ionizable lipids that have increased biodegradability, low reactogenicity, allowing redosing. Number two, decorating these particles with targeting binders, antibodies or antibody fragments in order to enable the reprogramming of immune cells in vivo and further detarget the liver. And number three, rethinking the chimeric antigen receptor architecture as well as optimizing the mRNA chemistry to maximize the expression of the chimeric antigen receptor on effector cells in vivo. And we achieve that through optimizing 3', 5' UTR, RNA chemistry itself, through using 5,1 and methyl pseudouridine, and other aspects, including codon optimization, fitting, proliferating, or effector T cells. These targeted lipid nanoparticles decorated with anti-CD5 or anti-CD8 antibodies, reprogramming CD4 more predominantly or CD8 positive T cells, and loaded with anti-CD19 chimeric antigen receptor in this case, could effectively reprogram T cells in this humanized animal model, mouse model, in order to express the CAR rapidly within 48 hours post-infusion, leading to ablation of NALM6, in this particular case, leukemic cells. During the last 2 years, though, several reports showcase the profound clinical activity of conventional ex vivo viral anti-CD19 or anti-BCMA chimeric antigen receptor T cells in B-cell involved autoimmune disorders. Owing to the achievable target product profile utilizing a targeted lipid nanoparticle format for our transient mRNA CAR payload, we prioritize B-cell involved autoimmune disorders for our first line of products at Capstan. How does that really work with an in vivo transient and tunable chimeric antigen receptor format? This is depicted in this slide, as you see here. On the left-hand part of the slide, the chimeric antigen receptor is expressed of the mRNA delivered through targeted lipid nanoparticle in vivo. Then these chimeric antigen receptor engineered T cells have the capability to rapidly engage cognate B cells in the microenvironment, secondary lymphoid organs, tertiary lymphoid structures in inflamed organs or bone marrow, and induce their ablation. This process happens very rapidly within days post-treatment. The treatment can be envisaged as a series of administrations several days apart in order to maximize the engineering of T cells. Post-global B-cell ablation, especially involving post-switch memory B-cells where autoimmune repertoire resides, this chimeric antigen receptor of mRNA naturally clears within days, allowing start of repopulation of the normal B-cell lineage from precursors in the bone marrow, hematogones, recapitulating actually the B-cell population, largely dominated initially by naive B cells without the autoreactive B cells. That leads to durable drug-free responses, and that's the target that we would like to achieve in this case. So you could appreciate that by transiently reprogramming the immune system, utilizing a tunable approach based on mRNA, in principle, one can achieve a durable response if one can repopulate or induce repopulation of the immune repertoire with normal cells devoid of autoreactive cells. I'm pleased to share with you the characteristics of our lead product, which is called CPTX-2309. This is an anti-CD19 chimeric antigen receptor utilizing an optimized mRNA format and CAR architecture that is an all-human CAR that is formulated in a targeted lipid nanoparticle using our proprietary ionizable lipid associated with increased biodegradability, reduced reactogenicity, and decorated with an anti-CD8 monoclonal antibody in order to reprogram effectively in vivo CD8 positive T cells. There are a couple of important features associated with this product. Number one, it targets CD19, and there are two reasons for that. First, we would like to utilize a validated target in order to guide the optimization of this platform technology in early development, and then develop a broader portfolio of payloads fitting various disease indications. Secondly, CD19 can be a very useful target to induce immune reset, drug-free durable responses in autoimmunity, since it is expressed not only on short-lived plasma cells, naive B cells, but also on post-switch memory B cells where the autoimmune repertoire really resides. And the second important feature of this product candidate is the fact that it is aimed to reprogram cytolytic lymphocytes and not co-opt T helper CD4 positive T cells that are responsible for massive cytokine production. We feel that this is a potential competitive advantage for this type of products as it may induce lower cytokine release in vivo. CPTX-2309, which is this anti-CD19 chimeric antigen receptor formulated in CD8 decorated targeted lipid nanoparticles, is very effective in rapidly reprogramming CD8 positive T cells in humanized mouse model and inducing within 24 hours ablation of B cells, both in blood, but especially important in tissues such as spleen. Just in 4 weeks, we will release at the annual ACR meeting in Washington, D.C., other evidence in support of CPTX-2309. Importantly, non-human primate data showing profound ablation of primary B cells in NHP, followed by repopulation with naive B cells reminiscent of immune reset. Secondly, we will also show data that speak to capability of CPTX-2309 to reprogram in vitro patients' T cells. All of this information, mouse data, other rodent data that we'll be publicizing, as well as non-human primate data, in vitro evaluation of engineering of T cells from patients, will be utilized to generate a springboard for future clinical development of CPTX-2309. We have line of sight to initiating this trial soon. Beyond CPTX-2309, we are very interested to develop a best-in-class portfolio for B-cell-involved autoimmune disorders. Additional targets are shown here, such as BCMA, that is expressed on long-lived plasma cells. We are also very interested to collaborate on a broader range of programs with targets such as CD20, as well as FAP, fibroblast activate protein, as well as other targets in oncology as well as non-oncology indications. However, what's beyond chimeric antigen receptors? And of course, our entry will be in vivo CAR, and we are very excited to bring together the unprecedented efficacy of chimeric antigen receptors with the scalability and tunability of the platform that I described. But the possibilities are really limitless. And you see on the left-hand part of the slide here a concept of transiently reprogramming a cell or tissues with payloads such as RNA, mRNA. But on the right-hand part of the slide, what you see here is the concept of formulating and delivering gene editing systems, base editors, prime editors, RNA writers, and the like, which are aimed to more permanently or permanently actually change the genetic makeup of a cell, such as a stem cell. So we really see applicability of this type of in vivo targeted technologies in three major areas. Number one, in non-oncology indications, but not only in B-cell involved autoimmune disorders. Also with applicability in fibrosis, regenerative medicine, infectious disorders, and even senescence and metabolism. And secondly, we also see the possibility of applying this type of technology to oncology indications in this format of a transient mRNA payload, perhaps in earlier stage minimal residual disease where T cells are more fit, in context of combination approaches, including in vivo reprogramming or co-reprogramming of other immune cells, or co-delivering biological response modifiers or checkpoints utilizing TLMPs, addressing oncology indications associated with very high efficacy bars, such as solid tumors. And beyond that, of course, addressing needs in the area of genetic disorders through formulating and delivering payloads, such as gene editing systems. So altogether, we are seeing a bright future for in vivo reprogramming of the immune system to maximize really the potential of immunotherapy and achieve objectives that are currently not achievable with other treatment modalities, such as cell-based therapy, such as recombinant proteins, including T-cell engagers and cytokines and the like. So with this, I would like to thank you very much for the attention, and I believe we have just about 1 minute if there is any question you may have. Thank you.